programs. So we're almost 2 decades into this activity. This morning is all about students and the work that they've done over the summer. And so I don't want to make very long remarks, but I want to congratulate the 7 candidates for ASP that will present today. We look forward to hearing your presentations. The way this format works is that students will present orally for 15 minutes, and they'll be followed up with a question and answer period that will last about 10 to 15 minutes. So these are about 30-minute slots. We will ask student presenters to come in to the room when it's their time to present. And we're going to begin with our first volunteer, the one student that actually volunteered to go at any time in this morning activity, and that is Ellie MacMullan. So Ellie, the stage is now yours.
Thank you everyone for coming to my talk on how tissue engineering is a solution to the organ shortage problem. So this graph illustrates the discrepancy between the number of organs needed and the number received for the year 2020 in the United States. There are a variety of reasons why someone might need a kidney or a tissue transplant. One can be various forms of trauma or cancer or various diseases like coronary heart disease, type 2 diabetes that cause end-stage organ failure. In fact, one of the ways to minimize this gap between the light blue and the dark blue is to find treatments for those diseases that stop them from getting to the point where a transplant is a necessity. The other way to minimize that gap is to increase the supply of organs available for transplant. And that's where tissue engineering can come in, because it can do both, decrease demand and increase supply. Put simply, tissue engineering is the use of biomaterials to create functional tissues in a lab. You can think of biowings as the building blocks of these tissues. Biowings are typically types of hydrogels, which means they are polymers that can retain a lot of water. In some cases, they're liquid, like the one seen here, and then they undergo certain processes and become a more solid, stable structure. Another essential component of these biowings are cells. The cells in tissue engineering are always taken from the patient. This part is very important because even if someone is fortunate enough to receive, say, a kidney, they might still face rejection, which is where the immune system will attack those cells for being foreign. But if the cells come from the patient, that reduces the risk of rejection because there's nothing foreign there. The cells used are typically stem cells, and there's 3 types listed there. The most commonly used in tissue engineering is induced pluripotent stem cells, which can be taken from any tissue in the body and using genetic manipulation can be reverted back into stem cells, which are then differentiated into the desired tissue. The third component of these biowings are scaffolds. Scaffolds are like the recreation of the extracellular matrix or the microenvironment that cells exist in. They can be purely structural, or they can have biofactors that help the cells grow and communicate. They can also be artificial, like the graphene scaffold seen here. If they are artificial, the researchers choose which factors and nutrients are put into the scaffold, or they can be natural using a technique called decellularization. With decellularization, a tissue like the one seen here is removed from the body, and then the cells are then removed from that tissue. What is left is this spiderweb looking thing, which is actually a decellularized extracellular matrix. Those scaffolds are then seeded with the stem cells in the patient, and then that is what is grown in the lab. So those biowings, which is a combination of those scaffolds and those cells, are then 3D printed into whatever structure the researchers decide. There are 4 main types to do this. There's extrusion, lithography, spheroid, and laser-assisted bioprinting. Spheroid and laser-assisted are not very common because they're very complicated and there's a lot of disadvantages. So for the sake of time, we'll only talk about the 2 main ones. The first technique is extrusion bioprinting. With this technique, the bioink is in a nozzle and is pushed out in a continuous filament into whatever structure using pressure. As you can see here, typically the scaffold is placed first. Right here they're using artificial scaffold, and then the cells are placed next. You can also see that there's 2 different biowings here with different types of cells. This is actually a major benefit of extrusion bioprinting because it's super realistic, because the tissues are not made up of one type of cell. For example, liver tissues are about 80% hepatocytes, but 20% other different mixtures. So to be as realistic as possible with these organs, different types of biowings with different cells need to be used. Other advantages of this technique include accessibility, affordability, the achievement of high cell density organs, and a variety of bioinks can be used because it can be cross-linked or hardened using physical, chemical, or light methods. Really, the only disadvantage with this technique is that since pressure is used to push the bioink out of the nozzle, it can lower cell viability down to 40%. This is combated using larger nozzle sizes, but then that sacrifices high resolution and fast printing speeds. The other common technique of 3D bioprinting is lithography. There's two types, stereolithography and digital light lithography, and this figure here shows the digital light one. With this technique, one layer of biomaterial is placed, and then is photo cross-linked, which means kind of hardened using light, and then the platform it's on moves down, and another layer comes, and then that is cross-linked, and so on and so forth. The fact that the bioinks must be photo cross-linked is actually a major disadvantage of this technique because it really limits the number of bioinks that can be used. Also, the light used, UV light, can damage cell DNA in the cells. Other disadvantages include really slow processing times, and unlike extrusion bioprinting, only one cell type can really be used, which isn't super realistic. On the other hand, this technique can get really large, complex structures with high resolution, which are what organs are. So now that the kind of overview of tissue engineering is done, I want to talk about exactly how it's going to solve the organ shortage problem. So I mentioned earlier that one of the ways to do that is through disease models, so decreasing the demand. So this particular disease model is a cancer disease model. So what they did was this brain was 3D printed using extrusion bioprinting. It also has in it macrophage cells, which are part of the immune system, and glioblastoma cells, which are a type of cancer. And what these researchers discovered was that the glioblastoma cells kind of convert the macrophage cells into these malignant glioblastoma macrophage things that help the cancer progress in the brain. So the macrophages are doing the exact opposite of what they were intending to do. Instead of stopping the cancer, they're helping it progress. By this increased understanding of how this disease works, they can work on treatments and hopefully eventually cures that can help stop this cancer. The other way tissue engineering works to decrease the demand is through organ on a chip, which are primarily used for drug treatments. This is an organ on a chip. It's about the size of a USB drive. Inside those red and blue channels are actually whatever cells the researchers want to be studying, as well as channels for any nutrients that they want to flow through. To give some background, drug testing usually takes over $1 billion and between 10 to 15 years for approval. That's because the preclinical studies are primarily done on 2D cultures or in animal models, which aren't an accurate reflection of human physiological needs. So a lot of them pass the preclinical trials and move on to human clinical trials where they fail. Organ on a chip is working to solve that by being basically a simulation of what it's like in vivo or in a living body. You can see here, this is the entire system of what is used with the organ on a chip. So there's a heater there to make sure the cells are at the right temperature, because think about when you have a fever, you're pretty miserable. That's because your cells only work on like a high level within a very specific temperature range. There's also all of these pumps here with these tubes going in. That's making sure those cells are getting the right nutrients. Basically anything that would be flowing through and reaching the cells in the body will be done using this. Those tubes are also used to pump such as the drugs through if it's being tested for toxicity. And many models of this have been used. They've created lung on a chip where they 3D printed a cardiovascular tissue and then a membrane and then lung tissue to mimic what it's like in the lung. Some researchers also 3D printed a actual blinking eye on an organ on a chip. And this comes from a radiation experiment where they were testing the effectiveness of a drug to protect gut intestinal lining from radiation. So in this first column here, that's the control group. Those cells were not exposed to anything. As you can see, that's what they want it to look like because it's fairly healthy. This next column right here with the cells that were exposed to radiation and nothing else. That green is showing all the radiation damage. They discovered the radiation did things like increase the reactive oxygen species, cause DNA fragmentation, and an increased number of cells, including apoptosis. This third column is the cells that were treated both with the drug, which is the DMOG, and radiation. As you can see, those cells look a lot like the control group, which means they are still fairly healthy. There's no damage to be seen, at least in this graph, and the drug was effective. So these organ-on-a-chip models and the disease models are how tissue engineering works it from the demand end. So let's get into the supply end, which is the creation of the tissues and the whole organs. This first study is the creation of a personalized cardiac patch. Not only does this patch match the patient on the cellular level by using the patient's stem cells, it matches them on the biochemical level by using a decellularized extracellular matrix. It also matches this patient on the anatomical level. What these researchers did was they took a CT scan of the patient's heart and found where the major blood vessels were. They were able to 3D print those blood vessels in the exact way as it showed up on the CT scan. They then added a few more smaller blood vessels just to make sure there's complete oxygenation of the blood. And while this tissue was not implanted back into the human body, it did undergo a variety of structural and functional tests that it all passed over a course of, I believe, 3 weeks. This next study was the creation of a small-scale replica of a human heart, as well as a solution bath that can support the structure, a large structure. So the first step in any creation of an artificial tissue is to design it on the computer. And actually this part is really simple because there are already huge databases with all of these organs designed, and some of them already have all the optimal parameters for 3D printing, such as the liver. So really all the researchers have to do is go in there and select heart and then put in the dimensions they want. The next step is to the next step is to put all the bioinks together, make sure the scaffolds and the cells are all right. But after that, it is 3D printed, typically in a solution bath, to make sure the organ maintains its shape before it can be solidified into a stable structure. This next step is to remove the organ from the solution bath and put it through a variety of tests. So this one was a structural test where they injected the left and right ventricles with blue and red dye. As you can see, it's holding up. This is a very good thing because if the transplant heart is leaking blood, they're going to need another transplant or, at least another surgery. And then this last one is just imaging to show what it's like inside of that heart, and you can see those orange are the blood vessels going through the heart. So going quickly back to this solution bath right here, this is actually what this heart was created for, to test that, because previous solutions that were used could be used for smaller structures like this tissue, but when it came to the larger structures, getting it out put so much stress on the organ that it either lowered cell viability or the organ just kind of fell apart. So this particular solution was used because it can be cured in a variety of different temperatures. It can allow for free-form printing, so anything can be printed in it, and it's a safe, controlled method of removal, where actually that solution bath is, in a way, is melted away from the organ. So there's no need to put any stress or pressure on the organ. So even though this heart and that tissue wasn't implanted back into the human body, that really doesn't matter. Because even the research that has gone into tissue engineering has proven invaluable, whether it's the disease models that I mentioned, or the organ on a chip that are being used for treatment, or it's the creation of the solution bath, or different 3D printing methods that can solve all those problems and have no disadvantages. You might have noticed, or maybe you didn't, that a lot of these studies happened in 2019, 2020, 2021. This is really in the beginning stages of this process, and already so much has been achieved. Tissue engineering and biomedical engineering in general has just been called one of the most promising and fastest growing fields, not only in science, but across all disciplines. That's why I expect in the next decade, personalized tissues, not only for the heart, but for skin, liver, kidney, will probably be in human clinical trials, and very soon after that, I expect in standard treatment. I actually believe skin has been used before in the human clinical trials. And after that, the more complicated whole organs will follow fairly quickly. So although tissue engineering isn't the solution for the organ shortage problem at this exact moment, the potential of it is enormous, which is why I believe it is the solution of the future. Thank you.
Okay, we're going to open this up to questions now.
I've got 20.
Good, because I'm a history major.
Before my list? Please jump in, anybody. I do have a bunch of questions. Don't panic. They're genuinely questions of interest. Actually, I'm gonna start with a comment. which is, I love your optimism, but I do just want to remind you that gene therapy was also the promise of the future, and 30 years in has been unsuccessful until about 2 years ago with CRISPR, so let's temper our optimism.
But CRISPR has happened.
But it took 30 years! Like, we thought it was going to be the next day.
I did say you would need to give it some time.
I'm worried for you when you're working your doctoral work. I don't want you to be depressed like I was when it all didn't go the way I thought it would. All right, I guess my first question is, I'd like to go to a basic science question that kind of underpins what you're talking about. So you talked about how the glioblastoma cancer cells, and for anyone that's not aware, like that's a pretty nasty cancer. Like it's one of the ones you really don't want to be your diagnosis. So for the glioblastoma, in that study that you talked about, it impacted and changed the macrophages, sort of turning them essentially into cancer cells. Can you give us a very brief explanation of at least one mechanism by which they might be able to do that, some of the, sort of the underlying cellular function? I mean, I'm going after AP Bio here. This isn't meant to be like a mystery question. I just want to see if you can give us any of those underpinnings. You don't have to give us all of it, just like a little bit to explain how cells might become cancer cells. I know. I'm worried for you when you're working on your doctoral work. I don't want you to be depressed like I was when it all didn't go the way I thought it would. All right, I guess my first question is, I'd like to go to a basic science question that kind of underpins what you're talking about. So you talked about how the glioblastoma cancer cells, and for anyone that's not aware, like that's a pretty nasty cancer. Like it's one of the ones you really don't want to be your diagnosis. So for the glioblastoma, in that study that you talked about, it impacted and changed the macrophages, sort of turning them essentially into cancer cells. Can you give us a very brief explanation of at least one mechanism by which they might be able to do that, some of the, sort of the underlying cellular function? I mean, I'm going after AP Bio here. I'm not sure, this isn't meant to be like a mystery question. I just want to see if you can give us any of those underpinnings. You don't have to give us all of it, just like a little bit to explain how cells might become cancer cells.
Well, I believe how this particular happened was that the glioblastoma cells were giving off like signals to the macrophages that I'm not exactly sure because this scientific papers, but I believe that is what happened.
I'm not looking for the specifics of this. I'm really looking to see like the general sense of it. I think you're right on the right track.
Yeah.
So cell signaling and what's that going to change in those macrophages that might lead them into being cancer? Like what's the hallmark of a cancer cell?
That like it just replicates over and over and over again.
Perfect. So put those two things together. What's that cell signaling doing to the macrophage?
The cell signaling is making the macrophage produce more and more.
So it's disrupting the
mitosis.
Yes, thank you. Great. All right.
It's been a few years
Just want to make sure that we don't get so into the cool stuff that we forget the basic science. Awesome. And I think that's important. The reason why I wanted to go after this question is because one of the concerns for biomedical engineering is that when you create these tissues and organs, that depending on if you go the artificial route, scaffolding, and you're putting in biofactors, that you're not always 100% sure how they might influence surrounding cells, because those cells might end up messaging and signaling differently because we missed something, right? Like that's one of the challenges there. Cool. And then I guess my other question for right now would be, what do you think, if this was really going to become the mechanism by which hospitals treated patients in need of organs, what do you think that would mean in terms of how many locations, like the physicalness of it, like how far away could these labs be? How many of these labs are we going to need? How many people would we need? How much time does the patient have between diagnosis of needing that organ and actually being able to create that organ? And like, what do you think that might look like? There's no wrong answer. This is really like a philosophical, theoretical question. I'm like, well, what do you think that might look like for hospitals?
Well, unlike with normal like donations where getting that organ in time to the patient before the organ kind of like dies and is not useful, that's not really a problem because like it can, be transported, like, because it's an artificial system that's putting the nutrients through, so it can be transported. And yeah, so that's not really a problem. But are you also talking about, like, between the time the patient is diagnosed, will the organ be grown in time?
Yeah.
I definitely think, maybe, I'm not sure about the whole organ, but I think if it's just like a tissue transplant, I definitely think that's feasible, because they can do that fairly quickly. You know, if the stem cells are taken immediately, I don't think decellularization takes too long, and then it's just, and actually they can decellularize the whole organ. That's a fairly new technique though, so they don't even have to go through the whole 3D printing and curing process.
So we might only need like a few locations around the
Yeah.
country that could do this, and then they would just like ship them out essentially.
Yeah.
To where
That's what I think. So.
Alright, I will pause there. I have other questions, but other people should have.
Just to kind of add into this, what's the question? What's the timeline for growth on scaffold? Like, are we talking days or weeks?
Well, actually, I did see something that the scaffold can actually be put into the person and with the cells on it and then grow inside somebody. So there's that. So you don't even have to wait for, you know, it to mature outside. So that's one of the things. But also, yeah, probably a few days and then to get it really started. But the additional use of growth factors put in, even when it's already there, has actually been shown to really speed up that process as well.
Could you put to the hydrogel slide?
This one?
Yeah. So, just a kind of AP chem question here. So you said there is lots of water around this, right? So these are polymers. I think we had one class on polymers at the end of the year. But do you think these are hydrophobic polymers or hydrophilic polymers?
Hydrophilic.
That's great.
Thank you. I know that one. I was really
Last
worried when you
question
brought up
from me.
AP Chem.
So I had this hydrophilic because they will retain the water next to them, right? So, last question is.
That would have been my guess.
I had this burger recently, Beyond Meat burger. Now, we're growing meat outside the lab. Is that tissue engineering?
Aren't those like plant-based or I have.
But there are companies that actually make the meat outside, so it is actually meat, but it's not grown inside a cow.
It's kind of
grown
gross.
outside a cow.
It's kind
Have you
of
heard of this?
Yeah, I have not. It's interesting.
also plants are tissues. I just
Yeah.
wanted, like don't throw my plants off like that, Ellie. That's a great question too. How do you feel about that? Would you eat it?
I don't know. I gotta think.
Would you put one of
probably.
these organs in you?
Like one of these?
Yeah, one of these.
Yeah.
Then I guess you eat meat.
Then yeah, I guess I would have
Same.
to just even if I gotta stand by my own opinion. So.
I think we have enough controversy over GMOs, let alone artificial meats. Now, Ellie, I have a softball question. What has brought you into this area of inquiry? Why are you so interested in tissue repurposing and this bioengineering? Is this something you want to pursue later on in college?
I actually want to major in biomedical engineering in college, and if go to grad school and specialize in the tissue engineering. I discovered this just while I was looking into majors in the college search, and I just started reading on how tissue engineering works, and I just found it really fascinating because always my favorite part of AP Bio was all the biotech stuff. I didn't really like the ecology or all of the natural selection. I thought those were always the most interesting. So I just thought it was really fascinating when I came upon it.
Ellie, I have a question. Do you know down the road? I mean, I'm kind of thinking let's say, the advantage of this, right, is that it's your own tissue, right? But let's say down the road, someone genetically or just the way their body is built or whatever has naturally a bad heart or a weak, whatever, weak lungs or what have you. Do you think down the road they might be able to customize or tailor these organs so they would be even they would allow that person to live an even better life than before their organs stop functioning in a normal manner? Would there be a way to like supersize their functionality?
Yeah, well, these organs, they use like cells, but if the like full tissue is damaged, typically their tissue will not be used. It will be used maybe like, I think sometimes they use like animal tissues or just an artificial scaffold instead of getting like the natural one. So it should be like just a normal, it shouldn't have any of the like bad effects that they had because it'll be just a completely new organ. But
so
I don't.
For some patients, a replacement transplant may still be the better option than growing or not.
No, because well, these are replacement organs, even though it's created from their own. Because this.
Sorry, I meant like an old-fashioned donor.
Yeah.
With that, is there still a situation where that might be more advisable for the patient, or?
I don't think so, because it's really doing the same thing. But as I mentioned, because the cells are not taken. Say the heart is defectual, the cells are not taken from the heart.
Sure.
It's taken from any functional cells. Say like usually it's fat tissue. So as long as the fat cells aren't, messed up, then it should work the same way as just a typical normal traditional transplant.
I'm going to piggyback. I think he's asking a great question. So, and I'm just going to expand it a little bit. So, if the heart disease is not caused through life choices, but rather due to a genetic defect that would be present in all cells, including stem cells, what could be done prior to growing out the heart to correct that genetic defect that I mentioned earlier?
Yeah. I didn't think about that. Well, they can also use stem cells from other sources.
But what if you're stuck with the patient?
You have to use the patient.
You have to use the patient's stem cells. They have a genetic defect, and you want to create a new heart, but you got to fix it first.
Well, they can use like genetic engineering. Like the CRISPR you mentioned earlier can go in and fix those defects if they know where the defect is.
Correct.
That's part of the problem. They also have to figure
that's
it
sort
out.
of right.
Right.
I think what you're getting at.
I was afraid of saying it incorrectly, but my question essentially was, can you combine this with CRISPR? Could you, could ultimately those two?
I think so. I don't think they are currently
Right,
something that I know of, but this is not my area. But I think theoretically you can. I think that would be the power of it. I think the bigger problem is that the vast majority of organ transplants are not connected to genetic defects. I think they often are connected to environmental or lifestyle choices. And again, I'm saying that without actually having the data to back that. I just want, but I feel that that's my gut. Gut.
Yeah.
Radiation.
Well, they are already using genetic engineering to get the induced pluripotent stem cells into stem cells. So it's probably they can probably while they're doing that fix it. Hopefully.
It seems like it.
I guess.
It's a lot of